Yes, good evening, class. I welcome you to our revision sessions. Just note that we are doing today paper 2, section B, September 2024, Northwest Province. Like I promised you, I said that I'm going to do all the various papers and then make sure that all these various previous papers are what are revised comprehensively and are well covered. Without wasting time, I say that this is Northwest Province September 2024 paper. We are given the diagram that represents an animal cell in a phase of meiosis. A diagram that represents an animal cell in a phase of meiosis, and then that is it. Name structure. Name structure A. Name structure A. What is A? A, it is what? It is spindle fibers. I'm sure that we are aware that this A is what? Is spindle fiber. Others can say spindle thread. Other books or other sources can say spindle thread, spindle fiber, or thread. It's one and the same thing. It's one and the same thing. And then B, identify B. B is the cell membrane. B is the cell membrane which is given there. A is spindle thread or spindle fiber. B is cell membrane. And then our next question is saying identify the phase represented in the diagram above. Which phase is shown there? As you can see, we are having the chromatids, and then in terms of your understanding of meiosis, I'm sure that you can see this is anaphase what? This is anaphase 2. This is anaphase 2. This is anaphase 2. And then the question is saying give a reason for your answer. Why do you say that it's anaphase 2? Why do you say that why do we say that it's anaphase 2? It is because that the centromere the centromere splits, as we can see there, the centromere splits, and also the two chromatids are pulled to opposite poles, as you can see. The reason is because that the centromere split, the centromere splits, and the chromatids are pulled to opposite poles. They are pulled to opposite poles. That's the reason that we can see. So that is why we are saying this is anaphase 2. There's the centromere splits, and also the chromatids are pulled to opposite poles, as you can see from the diagram here that is showing on the board here. Which phase occurred before the one which phase occurred before to the one represented in the diagram. I once also said that this question, it is quite common and familiar. Metaphase 2 is the one that comes before. Metaphase 2 Metaphase 2 is the one that occurred before. Understand, to say the one that occurred before the one, before anaphase 2. Which one occurs first? It is metaphase 2. Please note that. And sometimes other questions might say which one follows it. So it is metaphase 2 that occurs what? Before this one. Before anaphase 2. So explain the consequences if the process causing variation did not occur between chromatids from the cell above. Number 2.25, 2.15. Explain the consequences if the process causing variation did not occur between chromatids from the cell above, what would happen? It will result in what? In similar gametes. It will result, the process will result in similar what? In similar gametes. It will result in similar gametes, and then offsprings, then offspring of the same what? Of the same genotype, of the same genotype and phenotype, offspring of the same genotype and phenotype will be formed, will be produced. They will be produced, and then what happens in that regard, and then will be produced, that will reduce chances of survival of those offsprings, and will reduce their survival chances. What happens is that if there is no process of promoting variation, the gametes will be produced that will be similar, and at the same time will produce offspring that are the same, which eventually reduces what? Which will also reduce their survival chances. At the same time, biodiversity. It reduces also what? Biodiversity. I'm sure that you know biodiversity. So what we are saying is that if variation is not there, it causes gametes of the same type and also producing offsprings that are the same in the genotype and phenotype, and then eventually will reduce survival of the organisms. At the same time, it is reducing what? Biodiversity. And then the next question, 2.16, how many cells will be formed at the end of the cell division? How many cells will be formed? How many cells will be formed at the end of cell division? I'm sure that you know that we will be having what? 4. I'm sure that we know that there will be 4 what? There will be 4 cells that will be formed there. And then the next question is saying tabulate differences. Tabulate the two differences between meiosis 1 and meiosis 2. I'm sure that you know that meiosis 2 is the same as what? As mitosis. This is one and the same thing. To say tabulate the difference between meiosis and mitosis. That is what the question is sometimes basically saying. So what we need to understand is that I can just try to make these answers fit here. I can say meiosis 1 this side and meiosis 1 this side, and then I will say what? I will say meiosis 2 this side. The difference is one, in this meiosis 1, crossing over takes place. Crossing over takes place in meiosis 1. In meiosis 2, does not take place. Crossing over does not take place. It does not take place. Crossing over does not take place in meiosis 2 class. That is number 1. Number 2, we also we need to understand that in meiosis 1, it results in 2 daughter cells. It results in 2 daughter cells. This one, it results in what? It results in 4 daughter cells. 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 4 daughter cel four daughter cells, meiosis II, it results in four daughter cells. We can also want to understand that in terms of meiosis one again, you need to understand that homologous chromosomes arrange they arrange at the equator in homologous pairs, and then in meiosis II, chromosomes arrange at the equator chromosomes are arranged at the equator in singles in meiosis II. One other difference again is that all chromosomes moves to opposite poles of the cell in meiosis I, and then in meiosis II, chromatids move to opposite poles of the cell. In meiosis I, it is the whole chromosome that moves to opposite poles of the cell. In meiosis II, the chromatids move to opposite cells of the opposite poles of the cell. That is one other reason or one difference that you should understand there. And then in meiosis I again, the chromosome number is what is halved from the diploid to haploid. In meiosis I, chromosome number is halved. It is divided by half. But while it's in meiosis II, the chromosome number remains the same. The chromosome number remains the same in what? In meiosis II. Those are some of the differences that you can add in regard to the differences between these two processes. Lamarck's theory of evolution is based on two laws or principles: the law of use and disuse, and the law of inheritance of acquired characteristics. Question 2.2. Question 2.2. So what is the question saying? Explain the law of inheritance of acquired characteristics according to Lamarckism. Explain the law of acquired characteristics, law of inheritance of acquired characteristics according to Lamarckism. Question 2.21. So it actually explains modifications that are brought about by frequency of use or disuse, which are transmitted to offsprings in terms of acquired characteristics. We are saying it actually explains, it identifies modifications or changes. There are changes in this case that are brought about by frequency of use, changes brought about by frequency of use, or disuse, frequency of use or disuse, which are transmitted to offsprings. Which are transmitted to offsprings. So acquired characteristics are some changes that are coming as a result of the use or disuse of certain body parts, which are able to be passed on to offsprings. That is the principle or the law of use, the law of inheritance of acquired characteristics, according to Lamarckism. Next question, 2.2.2. Why Lamarck's ideas are not accepted in science of community? Why are they not accepted? One, Lamarck's theory, the reasons are such as those ones. The theory is too deterministic. Too deterministic. Whereby to say organisms do not change because they want to change. Organisms do not change because they want to change. That is one thing that is happening in this regard. But Lamarck said that organisms are changing because they want to change. So that is one of the shortfalls of Lamarck's theory. And also another point is that there is also one other point which also need to understand is that characteristics acquired by organisms through use or disuse do not affect the organism's genetic makeup, therefore cannot be what? Cannot be inherited. As number two, acquired characteristics, acquired characteristics cannot be inherited. Acquired characteristics cannot cannot be inherited. But according to Lamarck's theory, he said they can be inherited. Hence, we are saying is difficult. They do not affect the organism's genetic makeup. These characteristics cannot be inherited because they do not affect an organism's genetic makeup. So they cannot be inherited. So these are the reasons why the theory was what was declined. It is too deterministic, and also acquired characteristics cannot be inherited because they do not affect an organism's genetic makeup. They do not affect an organism's genetic makeup. Hence, those are some of the factors that makes the theory to be what? That which made the theory to be rejected as well. And then, next question. There are 4 blood groups in humans. That is true. Blood group A, blood group B and O, and as well as AB, as are the recorded phenotypes. Explain why blood groups are controlled by multiple alleles. Explain why blood groups are controlled by multiple alleles. Explain why blood groups are controlled by multiple alleles. So we are saying question 2.3.1. There are 3 different alleles that control inheritance of blood groups. That is the fact there. Because there are 3 different alleles. There are 3 different alleles that control inheritance of blood groups. That controls or that control inheritance of blood groups. That controls inheritance of blood groups. That is the point there. There are 3 different alleles that controls the inheritance of blood groups. That is one thing that we need to understand. 2.2.1, 2.3.2. How many of these alleles can an individual inherit in the genotype? How many of them? A person in a genotype can only have 2. Please take note, we are saying how many of these can an individual inherit? An individual inherit in the genotype. We are not saying how many blood groups control the blood groups. We are saying a single genotype is only controlled by what? By 2 alleles. That we must be able to understand it. Explain the role of blood grouping in paternity testing. It is actually pertinent in terms of question 2.3.2. Explaining the role of blood grouping in paternity testing. It is used to exclude a man from being a child's father. That is what it is in regard to what it is used to exclude. It is used to exclude what? It is used to exclude a man it's from being a child's father. That is how paternity is working in terms of blood grouping. It is used to exclude a man from being a child's father. That is what that's what a blood grouping works in this regard. Because we say that it can prove someone to be a man, but can sometimes not be 100% the father of the child. because many people can have the same blood group, which we can say we have got 100 of men, we have got blood group A or blood group A, B, or O. That is one thing that you should understand. Hence, it can only be used to exclude a man from being a child's father. Question 2.4, scientists wanted to determine what type of genetic disorder are found mostly in cats. And then to do this, they took 200 cats of the same breed from various genetic disorders. These are the ones which are shown here. The table represents the genetic disorders and percentages of each. Explain the concept of gene mutation. Explain the concept of gene mutation. Gene mutation, it is actually the changes in the sequence of nitrogenous bases in DNA molecule. 2. It's a change. It is a change in the sequence of nitrogenous bases. A changes in the a change in the sequence of nitrogenous bases in a DNA molecule. In a DNA molecule, which eventually, when this occurs, it can result in different formation. What it can result in different in the formation of different protein in this regard. So that is gene mutation, a change in the sequence of nitrogen bases in a DNA molecule. In simple terms, we can just put it in that way. And then state one way in which the reliability of the investigation was achieved. One way in which the reliability of this investigation was achieved, I said there was a use of a large sample size. A large sample size of 200 cats was used. Large sample size of cat was used. A large sample size of 200 cats was used. In this case, a large sample size of 200 cats was used. A large sample size of 200 cats was used, and also the cats were of the same breed. They used the same breed of cat. Used a same breed. They used the same. They used the same breed of cats. The same breed of cat was used, or a single breed of cat was used. Hence, we can say that the reliability was improved. Draw a pie chart to represent. We draw graphs. Here we need to draw a graph, but a pie chart to represent the data in the table above. So, in terms of drawing this pie chart, we are saying we must calculate the degrees first of each of these segments that need to be drawn here. So the pie chart must be drawn, which must be having a title. But before we go there, we must calculate. We must calculate the what? We must calculate these the percentages or the degrees of each of the representation of the types of genetic disorders that are here. So if we can add this one, 45 plus we are saying this plus 20 plus 20 is, if we can add this 20 plus that, we can get 100. So we are going to say that we calculate a fraction of this. This is equals to 45 over 100 multiplied by what? By 360 over 1, and then we can get the percentage 25 over 100 multiplied by 360. Also, we are going to see how much degrees are we going to get there. 20 over 100 multiplied by 360. I'm saying here we are trying to look at, or we want to get the size of each of the sectors that we are going to draw here. So if we calculate these, we calculate that by multiplying by 360, which is a complete revolution. So the first one, if we can calculate, we can get 260 degrees, 362 degrees. And then if we calculate this one, the second one, we get 90 degrees. And then if we can also put it on the calculator, the next one, we get what? 72 degrees. And then lastly, we get the last one, which is what? 36 degrees. These are the various representations that each of the genetic disorders are representing on the pie chart. So when we are drawing a pie chart now, I'm just going to draw to say that there must be a title. Draw a pie chart to represent the data in the table above. So is percentage of genetic disorders in case that is the title of my pie chart. Percentage of genetic disorders in cats. That's my title. And then I can just draw my pie chart, which is going to be a circle. Sorry. I can just draw my pie chart, which is just a circle. And then what we are saying is that diabetes is 65. I'm saying diabetes. You can even also just even give them as what? As percentage as they are. So we have got diabetes, which is got diabetes is almost like to say is almost more almost half of this, which we can say it can be like this, which is diabetes. 300, how much is it? 162 degrees. 162 degrees. That is diabetes there. And then we have got the next one, which is what? Which is polycystic kidney disease, 90 degrees. Which one? Sorry. Which is 90 degrees. And then in say 90. And then we have got the other one, which is deafness, which is 72. And then the smallest is what? Is 36. So I can say something like this. So here it is here is other, and then is what other is what is 36 degrees. 36 degrees, and then I'm saying 36 degrees, and then we have got deafness, which is 72 degrees, and then we have got polycystic kidney disease, 90 degrees, and then we have got what? 90 degrees, which is what? Polycystic kidney disease. Polycystic kidney disease. I'm just writing in short, but you can write it in full as you are writing in your book there. So what I'm saying is that these are our various degrees of each of the various what you call presentations or genetic disorders that are given there. So we are saying it's 162 degrees here, 162 degrees, which is given there, 90 degrees, 72 degrees, and 36 degrees, something like that. So our pie chart is given. But one key thing that you can do is that you can draw by calculating the degrees of each of the what? You can calculate the degrees for each of the segments that you are going to draw there. The degrees are calculated here by multiplying these by 360 over the total of each of the over the total. So we can as well draw them straightforward because here it is given as percentages. So if it is percentages again, we can as well define to know which one is bigger than the other. In this case, we are given as percentages. So even though we may not have calculated the degrees, but the diagram was still going to be drawn, and then it was still going to give us what? Uh, it was still going to give us the correct answers that are fine because we know that 25% is bigger than 20%, is bigger than 10%, and 45% is the biggest. That's how it is in terms of this. So this is how we were going to calculate these what? That's how we were going to calculate the different sectors there. The diagram shows protein synthesis. It shows this diagram shows the diagram photosynthesis. I'm sure this one again, it is also, please refer to the channel also. Refer to channel for more notes and more explanation again. I worked it out again there. Identify process V. Identify process V, which is process V is the process which is causing or which is giving to what? To the formation of mRNA. Identify the process. This is process V is what? Is transcription. This is transcription class. Process V is transcription. Is transcription. That is process V. And then identify two places where molecule X is found. Molecule X, where molecule X is found, where else is it found? So we are saying molecule X, which is what? DNA. Molecule X is our DNA. Where is it found? Identify two places where is it found. We are saying we need to understand that it is found in what? DNA is found in two places, which is found, it is found in the nucleus. It is found in the nucleus of a cell. It is found in the chloroplast of a plant. It can also be found where? It can also be found in the mitochondrion. That is very fine. We are saying where is it found? DNA is found in the nucleus. It is found in the chloroplast. It is found in the mitochondria. And then identify nitrogen base A. Identify nitrogen base A. Nitrogen base A. Where is it? Where is A? Identify nitrogen base A. I'm just checking where is A on the diagram there. Where is nitrogen base A? Our question does not show clearly. I'm not sure where is A, which they are referring to there. Identify nitrogen base A, or what are the, okay. Nitrogen base A, they are just saying, what does A represent as in terms of our nitrogen bases here? It's A, U, G, C. So what is A? A basically is adenine. I'm sure that we understand that A is what? Is adenine. Adenine, guanine, cytosine, and what? And uracil. I mean cytosine and what? And thymine. So what I'm saying is A is what? Is adenine. Describe the process occurring at Y. Describe the process occurring at Y. The process occurring at Y. Describe the process that is taking place at Y. At Y, it is the process of what? It is the process occurring at Y. I'm going to write it here. Process occurring at what? At Y, which is what? which is protein synthesis. So here we are saying one each there is each tRNA carries a specific amino acid. Each tRNA carries a specific amino acid. It carries a specific amino acid, and then what happens there? Our tRNA carries each tRNA carries a specific amino acid. When the anticodon when the anticodon which is found on the tRNA, when the anticodon on tRNA, when the anticodon on tRNA matches the codon on mRNA, matches the codon, when it matches the codon on mRNA, what happens there? Then tRNA brings the required amino acid. tRNA brings the required what? It brings the required amino acid. It brings the required amino acid to where? To the ribosome. And then what happens again is that the amino acid becomes attached to each other by what? By peptide bonds. Amino acid becomes attached to each other, by what? By peptide bonds to each other by peptide bonds to form the required protein. So this is what takes place. That is what is taking place there. It's a process of protein synthesis that you are supposed to explain here. The process of protein formation is actually what has to be explained in this respect of process Y, where we are saying our tRNA meeting, we are saying, don't forget, so next time they will say label those ones also. They are what? They are the anticodons. And then this is our mRNA. And then these are called the codons there. And then 3 nitrogen bases found on the mRNA are called codons. 3 nitrogen bases found on the what? On the tRNA are what? Are anticodons. And then we are saying the what? The amino acids that are brought here then. So please understand this diagram, class. We don't expect, or I cannot allow you to lose any marks there. You need to know that the 6 marks that are here, you get them all correct. Give the anticodon for the nitrogen bases. The anticodons for the nitrogen bases labeled Z. The anticodons for the nitrogen bases labeled Z. Obviously, like I said, Z is what? Is CCA. Z is CCA. I'm just going to illustrate to you guys. Z is equals to what? CCA. Like I said, these are what? These are the codons. And then their anticodons must be what? Must be G, G, GG what? GGU, because we are saying in terms of their pairing, we no longer have T, but we have got what? We have got U. So this is the codons, and then the anticodons are given there, which is GGU. Are we together there? I'm sure that you are getting this in regard to the complementary base pairing. That is what is happening. The answer is this one. Here I'm just extracting it for you to what? To see. I'm extracting CCA for you to just see where is GGU coming from. It is coming from the complementary pairing of the bases that you need to understand. If the third base on the molecule S changed to C, if the third base changes to C, uh, if the third base on molecule S changes to C, explain the consequences of the protein that will be formed uh, on S, on our S, on our S, this one, on our S, if the third base, 1, 2, 3, we are saying 1, 2, 3, 1, 2, 3. If the third base is going to change to what? If the third base going to change to C, explain the consequence to the protein that will be formed. Obviously, we are saying what would happen there. What would happen there is that in terms of that, the anticodon will change to what? Will change to, the anticodon will change as well. 2.2.5, the anticodon will change, the anticodon will change to what? Will change to, it will change to UAG instead of what? Instead of UAC. And then if that occurs there, that will code for different amino acid, which will code for a different amino acid, which will code for a different amino acid, resulting into a change in the sequence of amino acids. Resulting in a change of the sequence of amino acids. That's what is happening there. It's mutation. They are just asking you a question about mutation in this regard. If that changes, the anticodon will change also. Like I gave you, you just have to substitute the value C on the third amino acid and on the third nitrogen base, and then you obviously see that where there was it was A, now it is C. So if C replaces A, the change in the codon would affect also the representation of the anticodon. The table below shows the amino acid that corresponds to different DNA codes. You see, it shows the different amino acids. It shows the amino acid that correspond to different DNA codes. We have got the DNA code there, and then we have got the amino acids. Write down the sequence of from left to right of amino acids coded by S. Write down the sequence from left to right, of the amino acids coded by S. So our S, you just have to give this one like that. Our from left to right, our S, you are just going to mark them like this to say this is 1, 2, and then it's 1, 2, 3. So in this case, class, what you are supposed to do is that you are taking S in the order of the nitrogen bases that are there, and then you are supposed to do what? You are supposed to uh, give the representation of what are those amino to give the write down the sequence of from the amino acids coded by S from left to right. So I'm saying this can give you to say that this is the DNA, this is the DNA, and then you are supposed to write them in their order. So what is happening is that you are looking at this glycerin, arginine, and methionine. So when you look at them, looking at the order of those of these, you are going to code to say these matching to the S, matching to the H, matching to our, match them to our S molecule or most S structure there. What is happening there is that you will see that what is going to happen is that you will see that we will start with what TAC, which is going to match to what we are saying TAC, AUG, AUG, we have got AUG, we have got the next one is what? CCA, we have got CCA there, and then lastly we have got what? AGA. Lastly, we have got AGA as our S there. So what is happening is we are taking this. This is what? This is our mRNA here. And don't forget that when it is being formed, it codes to what? To DNA. So you are just giving to say what is the meaning of this. So TAU, we are going to have see that. I mean AUG, you see that this is going to be what? This is going to be TAC, which is methionine, and then you are going to see that this is methionine, and then you see this is CCA. CCA obviously is going to be GGT, which is represented by what? Which is represented by glycine. And then obviously the last one is going to be TCT, which is TCT is what? Arginine. This is the order where they are saying write down the answer, write down the sequence from left to right of amino acid coded by S. So I'm saying our S class, this is our S is that one. This is our S, which is represented by this. S is like this. This is our S here. This is our S structure class, this one. So the S structure is given like this. So we are just giving the corresponding DNA codes that are corresponding to the what? To GAUG, it is TAC. So TAC is methionine, meaning to say we are going to write methionine first, and then we are saying CCA is GGT, which is glycine, which are going to be this one. And then lastly, you are having what? Arginine, which is given as TCC, TCT, which is going to be given as that one. Hence, we are saying that's how you write what? The order of these nitrogen bases. Just quickly, I have the last one. Let's just get the last party. Give the term, define the term species. Species is a group of organisms sharing the same characteristics that are able to interbreed. It's a group. It's a group of organisms. A group of organisms that are sharing the same characteristics. sharing same characteristics, that are sharing the same characteristics, and then are able to interbreed and are able to interbreed to produce fertile offspring. That is a species. A species is a group of organisms that are sharing the same characteristics and are able to interbreed to produce a fertile offspring. We call that a what? We call that a species. Explain how the different species of the crabs could have evolved in South Africa, South America, and Australia. So I'm saying this question again, uh, you need to explain the geographic isolation. Geographic isolation is what you should explain in this case. You need to explain geographic isolation class in this case. Geographic isolation. The population were separated into different groups, which they were which they undergo natural selection, not being able to interbreed with each other uh, without which caused gene which there was no gene flow. Then the organisms, they were genotypically and phenotypically different, which would result in formation of different species, such that when they meet again, they cannot be able to interbreed. So I'm saying it is a question of geographic isolation. Please refer to channel again. It has been repeated quite often, this question. That's why you see I answered it quite often, this question. Refer to the channel of geographic isolation or forms of speciation. It's also written as speciation. Under life science grade 12, speciation, geographic isolation, then you look on that. It is well defined and explained, just to save our time again. Mention two reproductive isolation mechanisms that keep species separated. Reproductive isolation mechanism that keep species separated. What are those? Uh, they are quite a lot. We are saying one, uh, it can be a specific species courtship behavior, specific species courtship behavior, courtship, courtship behavior. Certain species, they have got specific exhibition that they do in terms of courtship. It can be breeding in different times of the year, breeding in different times of the year. It can also because of adaptation to different pollinators, adaptation to different pollinators. There are also another reason which we can also finally say that infertile offsprings, infertile offsprings, which is sometimes also called hybrid sterility. Some books will put it like that. So these are the different reproductive isolation mechanisms that can keep species separated. They can keep species separated. Yes, like I am saying, uh, let's just quickly check the next question. The diagram below represents the pelvic structures of and the ventral view of the skulls of three organisms. 3.2.1. These are the things. We write down the letter of the skull that shows the foramen magnum of the foramen magnum of what? Of Homo sapien. The foramen magnum of Homo sapien, which we are saying the opening at the back is what we call the foramen magnum, the opening at the back. So the Homo sapien, the letter of the skull that represent the Homo sapien, it is M. You can see that M is the one that represent what? That represent the Homo sapiens. M can represent the Homo sapien, and then explain your answer. In this case, the foramen magnum is in a more forward position. Uh, the uh, the foramen magnum is in a more forward position. Is in a more forward position, and then it is in a more forward position. Describe how pelvis. Describe how pelvis D is representative of a quadrupedal organism. To describe how this is a representative of a quadrupedal organism. It is D, the pelvis of D. This one. How can we say that it's a quadrupedal? How can we say it is what? It is quadrupedal. One thing which happens is that it is long and narrow. It is long and narrow. It is also long, narrow. It is long and narrow, even also lower. That is other points that we can see. You see, if you can check here, the length in terms of it is long, narrow, and lower also. It shows that it is what is quadrupedal. And then the next question is saying, describe two visible difference between the jaws of skull K, the jaws of skull K and L. Skull K, describe visible differences in skull K and what? And L. What are the differences there? This is skull K and L. As you look at them, the differences in terms of the jaws, what are the differences there? We can give these differences one, in terms of the differences, one, what we can say is that the canines of skull, canines of skull are the canines of skull K, they are what? They are short. They are short. Those are what? Well, for L, are what? They are long and pointed. As you can see, the canines of what? Of L, the canines of K are what? Are short and they are short. Well, L are what? Are long and pointed. The canines, as you can see, these are the canines that we are talking about. Here we are saying they are short. We can't see there. They are quite short. And then one other difference that is there again in terms of the skull is that the jaw of K is less progenious. The jaw of K is less progenious. That whereas that of M is what? while L is more. That is one difference again. And then thirdly, I can as well say that I can as well also that the plate of K is short and semicircular, is short and semicircular, is short and semicircular. The plate of K is short and semicircular, and then L is what? Is long and rectangular. I'm sure these are some of the differences that we can see there. These are quite notable differences that we can see from from the scales that we are dealing with or that we are referring to in this question. The scales that we are referring to in this question, we can agree that they like that. It is K is short and semicircular. K is short and semicircular. You can see it is short K. The plate is short and semicircular, but this one is what is long and rectangular. L is long and rectangular, as you can see there. So these are some of the differences that we can give there. And then the next question is the possible representation of human evolution. Possible representation of human evolution 3.3. The possible representation of human evolution. And then you are given all this. Name the type of diagram that is shown in on this one. It is a phylogenetic diagram. This is a phylogenetic tree. This is a phylogenetic tree diagram. And then another question is saying how long did Australopithecus afarensis become extinct? How long did Australopithecus afarensis become extinct? Australopithecus afarensis become extinct. We can look on that. Let's look on Australopithecus afarensis. Australopithecus afarensis. How long did it become extinct? So we are saying it started here and then it ended here. So you can drop it down and then you can see that this is Australopithecus afarensis. So what is it? It is what 3 million years ago, as you can read on the graph there. We can say 3 million or it can be maybe 3.1 or what. So I can maybe possibly say that it's 3.1 million years. 3.1 million years ago, identified two species that coexisted with Homo erectus, two species that coexisted. We are saying co means existed at the same time with what? With the Homo erectus. Two species that coexisted with the Homo erectus. We have to look for Homo erectus and two species that lived at the same time with it. Two species that coexisted with the Homo erectus. Homo erectus is here. Homo erectus, we have got, it existed same time with this one, which is the Homo habilis. Homo habilis, they existed same time with it. And then we can as well check which one also, the Homo habilis, they existed in the same time with them, the Homo habilis. And also, we also have to check again, coexisted. Okay, it also coexisted with what? Uh, it also coexisted with the Homo sapiens. I'm saying from here to there, is the Homo habilis, and then we also have the Homo sapiens on this party from here to there, which is also showing the what? The Homo sapiens. So it is the Homo habilis, the Homo habilis and what? And Homo erectus. They existed same time with what? With I mean Homo sapiens. It is Homo sapiens here, Homo habilis and Homo sapiens. And then question last, state one advantage of a large cranium capacity in Homo sapiens. One advantage of large cranium capacity is saying the large in terms of the skull. So the increase in the brain size to allow for what? For intelligence and also quick processing of information. It allowed uh, it allowed for large brain size, large brain size, which increased intelligence and quick processing on and quick processing of information and quick processing of information, which is an advantage in terms of the cranium size. And then calculate the time difference. Calculate time difference between the evolution of Homo erectus and Homo sapiens. So in this case, calculate the time difference. We are just saying the Homo sapiens and Homo erectus, Homo sapiens and Homo erectus, the time difference, Homo sapiens and Homo erectus. So what is happening here is that we are just saying the time difference between Homo sapiens and Homo erectus. This is it. This one is 0.2, and this one it is what? 2. I mean, it's 0.5 and also 2.0. So what is happening here is that we are just subtracting this to say 2.0, 2.0 minus 0.5 is equals to what? Is equals to 1, what? 1.5. 1.5 million years ago. And then describe how the fossil of the species in the diagram provide evidence of out of Africa hypothesis. How does that provide evidence of out of Africa hypothesis? This also refer also out of Africa hypothesis to the channel also. It has been answered somehow better as well. So the fossils of the Australopithecus and also Homo habilis are found only in Africa. Fossils of of Australopithecus and Homo habilis they were only found in Africa, were only found or are only found in Africa. The fossils of Australopithecus and Homo habilis are only found in Africa. That's one thing that we should understand again. And then the oldest fossils of Homo erectus and Homo sapiens are only found in Africa also. oldest fossils of Homo sapiens and the oldest fossils of Homo sapiens and Homo erectus are only found in Africa. Are only found in Africa. And then lastly, we can also say that the youngest fossils are found in other continents. Youngest fossils are, are only found in other continents. Youngest fossils are only found in other continents. That is also the explanation behind what out of Africa. Out of Africa, it is explained by this narrative. Question 3.4. Question 3.4. In rabbits, brown fur is dominant to white fur and long ear and dominant to short ears. A rabbit that is heterozygous for both characteristics is crossed with a rabbit that is white fur and short ears. So how many genes for the rabbits are considered in this crossing? There are how many? There are two, which is called dihybrid cross, which is called dihybrid crossing, and then uh stating the genotypes of the parents. The genotypes of the parents, we are saying are the parents are what? Is B. Is saying the first one is heterozygous for both parents, for both characteristics, which is BE, and also the other one is crossed with a rabbit with fur, and which is also recessive, for both characteristics. So that is the genotypes of this one. These are recessive alleles that you can see here. And then the next question, give the phenotype of rabbit that is dominant for both characteristics. The phenotype of this one, 3.4, it is going to be what? Brown fur with long ears. Brown fur with long ears. And then the next question is saying all the possible genotypes of the gametes of the heterozygous parent. Like I said again, refer also to notes, refer to channel again. I have got a question in terms of the dihybrid crosses and also other questions that are calculated in that regard. Please refer to the channel. So in terms of this one, the big question are the possible genotypes the possible genotypes of the gametes of the heterozygous parent. This, I always tell you that it must give you as many gametes as possible. So the gametes can be the dominant one gamete, you can have that one. We can also have the B and E. We can as well have the recessive B and also E there. We can also have a gamete that is what? That is B and E, which is recessive for both. So when we are looking at this, I'm saying there are quite possible gametes that can be produced there. There are quite possible gametes that can be produced of the genotypes of the gamete heterozygous. All the possible genotypes of the gametes of heterozygous parent. So you can see that we can have possible gametes such as these ones that I've listed there. I told you that the gametes must always have the different characteristics that are in question. We cannot have dominant B and recessive B in their own, or a dominant E and recessive E, but it must always have what? Both characteristics, E and B must always be there, not of the same characteristic. Hence, that will be an error. But the correct thing must always have both what? Both characteristics in the what? In the comment. Statement, those law of independent assortment, the law of independent assortment there, it says that alleles of different gametes, alleles, the alleles of different genes, the alleles of different genes, the alleles of different genes, they do what? Segregate, the alleles of different genes segregate, and then they segregate, and then into gametes so that they appear to the gametes in the different combination. So that they appear, so that they appear on different gametes in different combination. In different what? In different combinations. That's one thing that you should understand. It is actually a law that talks about the different movement of gametes, so therefore they can reappear or they can just reshuffle themselves. So what happens is that the alleles of different genes segregate, or we can say they move independently of each other into what? Into the gametes, so that they appear what? On the gametes in different combinations. They move independent of each other, so therefore they appear on different gametes in different what? In different combinations. That is what is happening in terms of this independent assortment that we are talking about. That's what exactly that's what exactly you need to understand. And then just quickly move on. The filial hyper disorder that is passed down through families caused by LDL bad cholesterol level by high to be very high. This disorder shows incomplete dominance, and then it is given by the following codes. So the question is saying, describe what is meant by incomplete dominance. What is meant by incomplete dominance? It means actually neither of the alleles are dominant over each other. Neither of the alleles, neither of the alleles is dominant over each other. Is dominant over each other. That is what you mean by what? By incomplete dominance. Neither of the alleles is dominant over each other. Therefore, it results in an intermediate phenotype which is obtained or which is produced, and then resulting in an intermediate. It results in an intermediate phenotype. An intermediate phenotype is produced when both alleles are present. When both alleles are what? Are present. Give evidence from the information above that shows that inheritance of the condition is an example of incomplete dominance. Evidence from there. And then in this case, if you can read, we can see that individuals with have a mild form of what of the disease. That is individuals. We have got individuals who are what? Individuals. We have got individuals with the genotype. We have got individuals with genotype what? With the genotype HN and also HS. These individuals have a mild form of what of the disease? Have a mild form of the disease. Um, the next question is saying a man heterozygous for the condition marries a woman who is normal, and then in a genetic cross show the percentage chance of them having children with severe condition. So what we just need to show here, I'm saying refer also to the what? Refer to the channel also. Refer to channel as well. It is quite calculated. So here we are just saying a man heterozygous for the condition marries a woman who has normal who has got normal stuff. So we are saying a man who is heterozygous marries a woman who is normal. So we are saying the man who is heterozygous, it means he has got the condition. So we say P1 phenotype, and then we are saying affected men. It is affected men. Affected men marries a normal woman. And then what we are saying is that it's an affected man who marries a normal man, and then this man has got the genotype. The genotype is supposed to be what? Is supposed to be HNHS, and then he marries a woman who is what? Who is HN. So that's the genotype there. And then we are saying meiosis, and then the gametes we are going to have HN, and then we have got HS, and then is marrying this one, which is going to be 1HN, because it helps the chromosome, the number of chromosome there. And then we say fertilization is this one, and then it's there. So we say HN, this one. This is just, we are taking the same way as it is. HN, and then is HN, and then this is HS, and then it's going with this what? It's going with this HN. Then we have got this, which is going to be the genotype. And then when we say that is the genotype, so the phenotype is the interpretation of this representation. So this is what one normal versus one affected. Child, one normal child, affected child. So the question is saying what? What does the question wants us to say? It's saying give the what? It's saying give the in the genetic, show the percentage chance. So it is 50%, which is 1 over 2 multiplied by 100 over 1 is to 1 over 2 multiplied by 100 over 1, which we are saying is going to say 50% chance affected is to 50% chance is 50%, 50%. So it is chance factor over total ratio in this case, guys, in getting percentage. In getting percentage is chance factor over total ratio and then you multiply by 100 over 1 in order for you to get what? In order for you to get the percentage. In order for you to get the percentage is chance factor over total ratio multiplied by percentage. Then you get the what? You get the answer that you want. Chance factor over total ratio multiplied by 100. And then you get what? You get your is equals to percentage ratio. That's the simple ratio. That's the simple formula that you should understand. So please, thank you so much for your time, and don't forget that these questions they are quite often, and we are actually preparing for the final exams. So please keep connected and keep uh, uh, following up, share, comment, and also uh, connect to others, and then we can do good. Thank you so much for your time, and don't forget to share to friends, families, and others so that all of us will get distinctions. Have a beautiful day and all the best.